in our discussions on antimicrobial chemotherapy, and today we're going to talk about how bacteria become resistant to antibiotics. And then we're going to segue into clinical microbiology, which aims to understand different microbes that are infecting patients in a clinical setting, because as we will see here and what we discussed last time is that sometimes it matters to know what microbe is infecting a particular patient. Okay, so you can make a targeted choice with your drug prescription. Okay, so the exam review is going to be March 15th from 6 to 7:30 in Olmsted, room 1208. Okay, is it 1208? Okay, for some reason I have 1204 stuck in my head. Okay, so we're going to talk about antibiotic resistance now, after we talked about the targets of antibiotics that are used for killing bacteria. And if you noticed, a lot of them are aimed at killing the organism. Okay, so if you think back to Darwin's theory of natural selection, this drives that population of bacteria to want to mutate, so they will be resistant to that particular microbe just through natural selection. That resistant to that particular antibiotic based on natural selection. Okay, so antibiotic resistance can come about through a mutation in the target. So for example, if the target is the 30S subunit of the ribosome, and it's a specific binding pocket in that, then there can be a small mutation there, so then that bacteria is no longer susceptible to that antibiotic. Or if it's DNA gyrase, for example, you can the bacteria can mutate that particular enzyme, and it won't be then resistant to or susceptible to nalidixic acid. Okay? So antibiotic resistance is a growing problem. So it wasn't until the 1940s that we really started using antibiotics in a clinical setting. And I do want to emphasize that they are one of the modern day miracles of the medical field, right? So infections that used to kill people are now easily treated by antibiotics. So it's not something that we should move away from in the medical field, but it's something that we clinicians now need to be cognizant of because of overprescribing antibiotics and creating this problem of antibiotic resistance in the bacterial population. So then these infections are no longer treatable by these drugs that we commonly use. Okay? And I want to emphasize that 80% of the antibiotics that are actually used are not prescribed to humans. It's used in farming practices for animals. So it was noticed that if you feed animals antibiotics, then they are healthier. Okay? And perhaps that's because they're in close quarters. It's not really 100% proven why they are healthier, but in fact, a lot of those antibiotics go into the feed of animals that are not sick. So it's just it allows them to become larger and healthier and produce more milk or meat for whatever their purpose is. Okay. And so there's a risk that those antibiotics can also then be discharged out into the environment. Okay, so they can get into waterways and things like that, and the bacteria that are in those waterways and everywhere are also exposed to those antibiotics. So there's a lot of selective pressure in many different environments for these bacteria to become antibiotic resistant. So this is just a graph. Okay, so this is 1940, so this is time on the bottom here, and this is percent of Streptococcus pneumoniae infections that are now penicillin resistant. Okay, so this is when penicillin was first introduced, and there were no reports of penicillin-resistant Streptococcus pneumoniae, and that's simply because people weren't really looking for that because penicillin didn't exist. But then as penicillin was being prescribed, and you can see here around 1979, there was a report in Spain that there was an antibiotic-resistant form of strep pneumoniae, and then if you look over time in the 2000s and the early 2000s here, you see many instances of reports of antibiotic-resistant strep pneumoniae, and now it's estimated that 80% of Streptococcus pneumoniae infections are now at some level of penicillin resistance. So we're going to talk about how does that even happen, right? So we talked about it being a part of natural selection, and I'm going to show a YouTube video about how that comes about. But these are the 4 basic forms of antibiotic resistance. Okay, so it's something that the bacteria have to physiologically change about themselves so they can be resistant to this particular antibiotic. So there are 4 major mechanisms we're going to talk about in this course. One is, like I said, alter the target. Okay, so you can slightly alter your DNA gyrase or your ribosome or whatever it is that that antibiotic is targeting. The second strategy is to degrade the antibiotic. So we talked about an example of that, which is our beta-lactamases. So those are enzymes that can degrade beta-lactam drugs like penicillin, and they're secreted out into the environment, and they're very good at chopping up the antibiotic, so it's no longer effective at killing the microbe. One is to modify the antibiotic. So once it's brought into the cell, it quickly changes it in some sort of chemical conformation so that it's no longer toxic. And one of the most dangerous strategies is this efflux pump. Okay? So this will pump antibiotics out of the cell, and a lot of these are very targeted. Okay? So that's a specific antibiotic or class of antibiotics that particular target has been altered to be resistant against. This would be, for example, our beta-lactamases. They target beta-lactams, but this pumping, these efflux pumps, can indiscriminately pump out antibiotics as they're toxic to the cell. And this job of this efflux pump is to secrete toxins. Okay? So this is where you can get multi-drug resistance very quickly if this bacteria has acquired an efflux pump that is designed to pump toxic things out of the cell. So arguably, this is the most dangerous form of antibiotic resistance. Okay, so this is just what I said, but in text form. So I'm not going to read through it. I just put it there so you can have it in for your notes. But again, I just wanted to say that this efflux pump can be non-specific in pumping antibiotics back out of the cell. Okay, so examples of destroying the antibiotic, our example is beta-lactamase, right? So this is penicillin. This would be the beta-lactamase, and this is the penicillin here, and it's destroying it, okay? And it can also destroy the analogs of penicillin. So we have a lot of things like amoxicillin that are analogs to penicillin, and these lactamases can cleave that beta-lactam ring regardless of the R group, okay? So they're very good at cleaving that ring. And what happens when these bacteria have these beta-lactamases is it makes the strain resistant, and it also lowers the penicillin concentration in the environment. And so that can kind of help other bacteria in the nearby environment as well, because it's degrading all that antibiotic. So the other ones might not necessarily be resistant, but they're benefiting. Okay? You can modify the antibiotic. So this would be, an example would be our aminoglycoside antibiotics. And so the bacteria can acetylate, so add an acetyl group to the incoming antibiotic, and phosphorylate it, they can adenylate it, depending on that particular drug, and then it can no longer interfere with the ribosome. And so that would then make that organism resistant to these aminoglycoside antibiotics who target the ribosome. Okay, so also altering the target, our example would be modifying the target enzyme of penicillin or like ribosome. Okay, so this would be the most common streptomycin resistance mechanism to alter the target, right? So we know that penicillins target the process of making peptidoglycan, and even more specifically, the transpeptidases and the transglycosylases. So if you can just alter that, the penicillin can't bind to it, okay? And then it won't interfere. Drug efflux is our multi-drug resistance exporter. So this is just an example of what one of those pumps looks like. And so you can see it spans the membrane, and it just pumps things out of the cell. And so it's essentially removing them from that cell, and you can confer multi-drug resistance that way. This can be very dangerous, like I mentioned, and so it's just a single thing that a microbe would have to acquire in order to be resistant to a lot of these drugs. Okay? It uses the proton-motive force, so it's not using ATP in order to accomplish this. So it's a very cheap form of energy, this proton-motive force, and so these bacteria can be very efficient at secreting these antibiotics. Okay, so I put this in this morning, actually, when it was like pouring down rain and hailing. I was like, I really need a graphic to explain this. So I'll upload this modified version of the slides after class. But how does this work, right? So we can conceptually understand that bacteria can be resistant. But how do antibiotics, the overuse of antibiotics, actually contribute to antibiotic resistance? So here's this graphic, and then I'm going to show a YouTube video, which I apologize in advance, it's very cartoonish, but I think it's good at explaining how these things happen, okay? So many bacteria are just naturally resistant to antibiotics. It's just part of their ecology, right? So if you think about what antibiotics are, they're natural products that bacteria make to outcompete each other, okay? So there is some level of natural resistance. So this resistance to antibiotics is not something that humans invented, right? It's just something that we have made worse, or made, made prevalent, okay? So you can see here that they've indicated through these pink bacteria that those might happen to be resistant to a particular drug, right? As soon as you start applying that selective pressure to that population, you start to kill all these blue ones that aren't resistant, okay? And then that leaves this environment wide open for these resistant ones to bloom, because you've killed off all the competing neighbors. And so then you would have a lot of these bacteria that are resistant popping up. And then even more, to exacerbate the problem, there's horizontal gene transfer in bacteria. So you can have this population, it might be just one species, okay? For example, Streptococcus pneumoniae or something like that. And then it can start transferring these genes that encode antibiotic resistance to their neighbors, okay? So then you can have more than one species that have this phenotype of being antibiotic resistant. So that's how that happens. I'm going to show this YouTube video. And please don't get mad at me. It's not for kindergartners, I promise. It's a TED Talk.
What if I told you there were trillions of tiny bacteria all around you? It's true. Microorganisms called bacteria were some of the first life forms to appear on Earth. Though they consist of only a single cell, their total biomass is greater than that of all plants and animals combined. And they live virtually everywhere. On the ground, in the water, on your kitchen table, on your skin, even inside you. Don't reach for the panic button just yet. Although you have 10 times more bacterial cells inside you than your body has human cells, many of these bacteria are harmless or even beneficial, helping digestion and immunity. But there are a few bad apples that can cause harmful infections, from minor inconveniences to deadly epidemics. Fortunately, there are amazing medicines designed to fight bacterial infections. Synthesized from chemicals or occurring naturally in things like mold, these antibiotics kill or neutralize bacteria by interrupting cell wall synthesis or interfering with vital processes like protein synthesis, all while leaving human cells unharmed. The deployment of antibiotics over the course of the 20th century has rendered many previously dangerous diseases easily treatable. But today, more and more of our antibiotics are becoming less effective. Did something go wrong to make them stop working? The problem is not with the antibiotics, but the bacteria they were made to fight. And the reason lies in Darwin's theory of natural selection. Just like any other organisms, individual bacteria can undergo random mutations. Many of these mutations are harmful or useless, but every now and then, one comes along that gives its organism an edge in survival. And for a bacterium, a mutation making it resistant to a certain antibiotic gives quite the edge. As the non-resistant bacteria are killed off, which happens especially quickly in antibiotic-rich environments like hospitals, there is more room and resources for the resistant ones to thrive, passing along only the mutated genes that helped them do so. Reproduction isn't the only way to do this. Some can release their DNA upon death to be picked up by other bacteria, while others use a method called conjugation, connecting through pili to share their genes. Over time, the resistant genes proliferate, creating entire strains of resistant superbacteria. So how much time do we have before these superbugs take over? Well, in some bacteria, it's already happened. For instance, some strands of Staphylococcus aureus, which causes everything from skin infections to pneumonia and sepsis, have developed into MRSA, becoming resistant to beta-lactam antibiotics like penicillin, methicillin, and oxacillin. Thanks to a gene that replaces the protein beta-lactams normally target and bind to, MRSA can keep making its cell walls unimpeded. Other superbacteria, like Salmonella, even sometimes produce enzymes like beta-lactamase that break down antibiotic attackers before they can do any damage. And E. coli, a diverse group of bacteria that contains strains that cause diarrhea and kidney failure, can prevent the function of antibiotics like quinolones by actively booting any invaders that manage to enter the cell. But there is good news. Scientists are working to stay one step ahead of the bacteria, and although development of new antibiotics has slowed in recent years, the World Health Organization has made it a priority to develop novel treatments. Other scientists are investigating alternate solutions, such as phage therapy or using vaccines to prevent infections. Most importantly, curbing the excessive and unnecessary use of antibiotics, such as for minor infections that can resolve on their own, as well as changing medical practice to prevent hospital infections, can have a major impact by keeping more non-resistant bacteria alive as competition for resistant strains. In the war against superbacteria, de-escalation may sometimes work better than an evolutionary arms race.
Okay, so that's antibiotic resistance in a nutshell. Okay, so what are some of the causes of this? So the video mentioned that, and I've mentioned this as well. So indiscriminate use of antibiotics for viral infections, for example, where they wouldn't be effective. So use of antibiotics when not needed. And also, just again to reiterate that most of the antibiotics that are used in this country are used in livestock, so 80% of them. And so that is, obviously not human use, but we eventually eat the products that come from those livestock, and they could potentially contain microbes that have acquired antibiotic resistance because they're in this high antibiotic usage environment. Also, so your doctor, when you get your prescription from your pharmacist, it tells you to take your drugs if you do need these antibiotics, and you should take the whole course of them. So it says, usually 10 days or something like that, and you start to feel better like around day 3 or 4, and you're like, oh, I don't need these anymore. So that can be really harmful, because what you've done is you have killed off all the bacteria that are very sensitive to the antibiotic, okay? And there might be some that have some low level of resistance, and if you don't keep applying that selective pressure, which is taking that dose of antibiotic when you're told to, then you will allow, and if you cease that natural selection by stopping your prescription, then you allow those somewhat resistant bacteria to then take over, okay? And so that's why your doctor and your pharmacist are adamant that you should take your entire course of antibiotics, and you should take it at the dose when they tell you to, right? So if you need to take 2 doses a day, you should take 1 in the morning and 1 in the evening, and be consistent with that. And the idea is to try to keep that concentration of the drug above the minimal inhibitory concentration in your body, okay? And then nosocomial infections. So these are infections that occur during hospitalization. So this can happen if you go in for something, like to get, like you broke your leg or something. Okay, you did not have an infection when you went there, but you acquired one because the hospital is like a breeding ground for microbes, because a lot of sick people go there. And because of that, there are also a lot of use of antibiotics, and there's a lot of antibiotic-resistant bugs that could be available to you and can be acquired by you during your hospital stay. Okay, very good example. This was my dad, actually. So he broke his kneecap, he fell down, and he had to go and get knee surgery. So he got that, and then they had to put like a metal wire in his knee to keep it together, and he acquired a MRSA infection in his knee. So he got osteomyelitis in his knee, and they, after some clinical microbiology that we'll talk about, they discovered that it was methicillin-resistant Staph aureus, or MRSA. Okay, so this, it's a big deal. How are we going to deal with this? And Indwelling medical devices are very good colonizing grounds for bacteria, so they actually decided to take out the metal piece that was in his kneecap because they decided, they weighed the risk and said it's better to take that out because these bacteria are forming these biofilms on this piece of metal. And so he had to have like 6 weeks of intravenous IV treatment of antibiotics, and they had to try different ones. And so they couldn't try the penicillins or the methicillins, so they had to go for vancomycin, and that was like in his arm for 6 weeks. And then he got a blood infection because something contaminated the IV, and it was something different. Okay, so then they did some more clinical microbiology where they showed that it was a gram-negative bacteria that was in his blood, and MRSA is a gram-positive. So he's telling me all these things on the phone. I'm like, well, I think that's good. Like, at least you don't have MRSA in your blood. It's something else. And so they had to give him a different antibiotic, and then he finally was cleared of all of those infections. But it took months, and it was crazy and scary, and lots of different IV bags strapped to him all the time. So all that because he was perfectly healthy and fell down and broke his knee, and then all of these things happened afterwards. Okay, and I'm not trying to scare you, I'm just giving an example, a real life example. Okay, so how can we prevent this, right? So we are smart, we're humans, we can, if there's a problem, we're problem solvers, and we like to solve these things that can be detrimental to the human race. So one thing you can do is you can give a drug in very high concentrations. So that's what they did with my dad. They were giving him like really high doses of specific antibiotics to try and get rid of that methicillin-resistant Staph aureus. You can prescribe 2 or more drugs at the same time. And so that can, if you don't know exactly what the resistance mechanism is, you can try to give 2 different drugs that have different targets, and then you can try and kill the microbe that way. Again, finish your prescription so you're not contributing to this problem, especially in your own body, of antibiotic resistance acquisition. And then use drugs only when necessary, so not to treat viral infections because that doesn't really help. And then obviously continue to develop new drugs, and we talked about an example of that when we talked about quorum sensing and the quorum sensing system as a way to develop new antibiotics that don't rely on killing the organism. So these would be ones that would confuse the pathogens. So you would create mimics of these quorum sensing molecules that bacteria use to talk to each other, and potentially jam that system. Okay. And also we talked about bacteriophage. So we know that bacteriophage are viruses that are specific to bacteria, and some of them are even specific to specific species of bacteria. And so wouldn't that be great if you knew that you had an infection, say Streptococcus pneumoniae, and you had this bacteriophage that was very good at finding it and killing it and leaving all the other microbes alone. So that is also another issue with antibiotics, is that some of them are broad spectrum, and so they kill a lot of things indiscriminately, and some of those are the good guys along with the bad guy, and that can create a dysbiosis in your body when you have killed off your microbes that are actually beneficial to you. So phage therapy is something that is alive and well in terms of the research pipeline. I don't know a specific example of phage therapy being used in a clinical setting, but again, I'm not a clinician, so I don't know everything about that. I do know that it's being field tested for plant disease control for Pierce's disease in grapevines, and it's showing some promise there. Okay, so what are more strategies to stay ahead of this? So you can chemically alter the antibiotic. Okay, so you can then turn to pharmaceutical sciences, and the chemists that are part of that say, okay, we have this drug, can we switch the R group, or chemically alter it in some way to where it's more effective or it's resistant to this beta-lactamase action or something like that. Okay. Again, you can use more than one antibiotic. And then you can also use what we call dummy compounds. All right, so this would be like a decoy that would bind more readily to the beta-lactamases. It would compete with the drug, is what I'm trying to say. Okay, so you would give these 2 compounds, clavulanic acid, for example, in combination with penicillin. And clavulanic acid is very good at binding to beta-lactamases, and so that would allow penicillin to go and do its job, where the beta-lactamases are busy trying to cleave the clavulanic acid. Okay? And I noticed that my cat was prescribed this when she had an infection. And so it was this combination of clavulanic acid and I guess penicillin, I'm not sure, that was given to her. And then, yeah, so let's see about that. Okay, so how does it develop? So the YouTube video mentioned this. So you can have random mutations, right? So that just happens during DNA replication, and that rate of that is very low. But if it's something that is beneficial to that organism, like drug resistance, it will persist in the population. It can also be acquired more rapidly through horizontal gene transfer. So genes are constantly being moved around within bacteria, either through conjugation, transduction, or transformation. So I just want to remind you that there can be two ways, right? So it can be just de novo antibiotic resistance, and that's through DNA replication errors that happen during that, and also through horizontal gene transfer. And then also de novo mutations can happen if the bacteria are exposed to certain mutagens that would mutate it in such a way that would be beneficial rather than deleterious. Okay, so the sources of resistance genes, so that, like I said, can be natural resistance. So there's a lot of bacteria that just are naturally resistant to drugs. And then you can have acquired resistance, okay? And this can be through mutation, either spontaneous or horizontal gene transfer. That can be from a plasmid, or it could be through other mechanisms, like moving chunks of chromosome around. And even transposons can carry anti-drug resistance here. Okay, so what's the future, right? So you guys are the future. So you're going to be discovering all of these things and pushing this field of science forward. So genomics is widely used today to discover new drug targets. So now we have the capability to sequence the genomes, the whole genomes of organisms very easily. And so that was kind of a black box before. Like we could look at certain genes and we could say, okay, we know this gene is here. We know it's close to this gene. We know that this other gene is probably absent. But now we can just sequence all of the genes and say, all right, here's what it looks like, and then we can create a model of what we think that bacteria's physiology is like, and then we can design targets for those and test them in a pipeline, and hopefully a high throughput pipeline where you could test things rapidly and to see if it would kill organisms or not. So using genomics to design compounds, okay? Or you could alter the compound, so that's where natural product chemists would come in to try and optimize the minimal inhibitory concentration. And you need to determine if the spectrum of that compound is narrow or broad. And then you also have to make sure through a series of tests that it's not toxic to animals. And one of those beginning tests, like we talked about, is the Ames test, where you're just using bacteria to see if these things have mutagenic properties. And then you also need it to persist in the body, right? So you don't want something that you ingest, like immediately gets degraded by your stomach acid, because then that's not going to be helpful in terms of a drug property. Okay, so that was all about bacteria, right? But we also have fungal infections that occur. Fungi are a little bit tricky because they're eukaryotes, just like we are. And so some of their targets are very similar to what we have. And so you kind of have fewer targets to choose from in terms of antifungal drugs. And sometimes they don't work as readily as antibiotics, okay, that would target bacteria. So a good example of this is athlete's foot, right? So that's a fungus that gets in between your toes and makes you itchy. And you can go and buy drugs from the pharmacy to put there, and you know that you have to keep applying that drug for weeks sometimes at a time in order to finally get rid of that infection, right? And so you usually have to have multiple repeated applications of antifungal agents because they're not as good at killing things as antibiotics might be. Okay, so there are some fungal specific structures you can target, like sterols in the cell membrane that we don't have, or chitin in the cell wall. We don't have chitin in our bodies. And so you can target that in fungi. But there's like efficient detoxification systems in fungi to inactivate lots of drugs. And so that's why you have to keep applying that foot fungal cream or whatever it is that you have bought from the pharmacy in order to finally get rid of those fungi between your toes. Okay, now viruses are another topic, all right? So viruses, they rely on our bodies to replicate. They're using our ribosomes and our translation machinery, so that's not something we want to target because then that would hurt us, right? So you have to specific target, and that can be difficult. And we have come a long way in understanding that, especially with HIV, trying to understand different processes that happen in those that don't happen in our bodies, okay? So it's very difficult to find selective toxicity because, again, I mentioned that that virus is using us to replicate, so you don't want to kill your own cells. Okay, one thing you can do is inhibit viral uncoating and release. So the example of that would be the influenza virus. So this drug, amantadine, inhibits viral uncoating. So it has to uncoat when it's in the cells in order to replicate. So this will prevent entry of the virus into the host cell. So therefore, it can't do its job of replicating. Zanamivir blocks neuraminidase, which is an enzyme specific to influenza, and that is preventing the release of the mature viruses. Okay, so there are two different drugs. One of them, let me see if I get this right. Yeah, one of them is Tamiflu, which you've probably heard of before. This drug here is commercially known as Tamiflu, and this Zanamivir that I just mentioned, they both block neuraminidase that is targeting the influenza virus. And so if you can take this Tamiflu at the specific timing of your infection, you might be able to prevent release of these mature viruses, and then your flu period will be shortened. Okay? And this is a relatively new strategy. We didn't have these anti-influenza drugs on the market in the not-so-distant past. Okay. And that's because influenza is an enveloped virus, okay? And so you can inhibit that uncoating and also release by messing with the envelope. Okay, so antiretroviral therapy. So there are some viruses that are retroviruses, and what does that mean? Okay, so they have to go through this process of creating DNA from RNA, all right? And so they use reverse transcriptase to do that. That's not something that we have in our bodies, so that's a good target, and it's unique to these retroviruses. And so one of our example retroviruses is HIV, okay? And so one of these drugs for HIV is to target this reverse transcriptase, okay, and to stop that from happening. Another drug is a protease inhibitor. So HIV has to process its proteins in order to be functional, okay? So you can inhibit that process through these two drugs that's inhibiting the specific HIV protease whose job is to process the proteins that are belonging to the virus. And then you can also have entry inhibitors, okay? So these block HIV from binding to the receptors on the cells of the human body and therefore preventing entry into the cells. So these are three types of drugs that are used, or drug targets that are used for antiretroviral therapy. Okay, so we're going to leave that now. We're going to talk about clinical microbiology. So we understand about antibiotics and antibiotic resistance, and sometimes we need to understand exactly what a patient is infected with. Okay, and so that is why we have this whole field of clinical microbiology. So there are many, many clinical labs across the country. Hospitals have their own clinical labs, and so when you're going and giving urine samples in a cup or whatever, that's clinical microbiology. They're trying to culture something out of you or do some sort of test on some bodily fluid that you have given them. That's clinical microbiology, okay? And the reason is like they want to, number one, determine do you have a urinary tract infection, for example? And if you do, like maybe they want to take it a step further and understand exactly what bacteria is in there, okay? And they also maybe want to understand if you have antibiotic resistance that is in that particular infection, okay? All right, so all these fluids that I talked about, they're common common ways of removing them or obtaining them from humans. So for urinary tract infections, obviously you need to collect the urine, so that comes out of the urethra, and you can collect that either just in a cup, or if you have to collect it through a catheter, that's also one way. There's also pus that you can take from infected, usually skin infections, and try and see if what's in that particular sample. Strep throat is a good way to diagnose that, is to do a throat swab, and then they put it in that little tube and they send it off, and they probably culture it on a plate and decide if you have strep throat or not. Sputum, so if you are coughing a lot and they need a sample of what is in your lungs, a good example of that is sputum, okay, and that's the thing that is dislodged from your lungs and put into this cup here. So that is one way that researchers can obtain and also study Pseudomonas aeruginosa, because a lot of it is dispelled in the sputum of patients that have that infection. This is a lumbar puncture, which looks horribly painful, but that's to get cerebrospinal fluid to see if you have any microbial infection in there. Okay, so you have to make sure when you're doing this that you're not contaminating the sample. Okay, and so for example, if you're getting a urine sample, they ask you to please, clean the area around that and then provide the sample to make sure that you're not contaminating just your normal skin microbiota or something like that, okay? Okay, so once you have this microbe, you want to identify it, and this is the process of clinical microbiology, okay? So you can use knowledge of the biochemistry of that microbe to determine if it is, in fact, that microbe and how it's different, perhaps from other microbes that you've gotten from other patients or other patients across the country, to determine if there's an epidemic of this particular infection, all right? And that just relies on, all microbes have different biochemistries, okay? So they have different metabolomic capabilities, and you can test that in a culture tube, okay? One easy way is to do a gram stain, right? So you can decide gram positive versus gram negative. So the example of the infection, blood infection, in my dad, they decided it was a gram negative and not a gram positive, which is what they were pulling out of his knee. So there were two different things going on. Okay, so that's a very easy way of trying to understand on a broad scale what's happening in the patient. Okay, and you can grow them at different temperatures and pHs and determine that way what biochemistries are underlying that particular microbe. You can also grow on selective media. So we talked about selective media, and that is media that's designed to allow only a specific group of microbes to grow. Okay, so if you're culturing out something and it doesn't grow on that particular selective media, or it does, you might say, okay, I'm dealing with this microbe because it grows on this specific media that only this microbe can grow on. Okay, and it also gets rid of all the other microbes that might have been contaminating that sample, and so that allows you to work with that microbe in isolation. Okay, you can also have differential media, so this would be an example where there's a color change that occurs in that media, and that will give you an indication that you're dealing with a specific microbe. Okay, so for example, E. coli colonies that can't ferment lactose, they will turn this media green here, right? And this up here is chocolate agar. It's not chocolate because you put chocolate in there, but that allows Neisseria gonorrhea to grow. And so if you think that a patient has gonorrhea, you can plate that sample onto this chocolate agar media, and then if those bacteria grow, you can be fairly confident that you have Neisseria gonorrhea growing out of that sample. And selective media is very good. So if you're trying to, for example, culture something out of a stool sample, which is very diverse and rich in microbial community structure, because you have a lot of microbes that are coming out in that sample, but you're trying to look for E. coli or something, that's why you can plate it on selective media that would allow that microbe to grow and also tell you through this color change that you're in fact dealing with that particular microbe. Okay, so moving on with this, so you can, like I said, do a gram stain, and that will tell you like which path to go down. It's kind of like a dichotomous key. But then you can do multiple tests at once, all right? And so this would be a strip of little cells that contain media, okay? Each media is different in each little cell, right? It could be different because it has a different pH. It could be different because it has a different carbon source. It could be different because it has a particular vitamin or lack of a vitamin in that particular sample. So what you do is you get your microbe of interest and you inoculate it into every single one of those little miniature culture tubes that are on this strip. And then you let it grow overnight or however long it takes for your microbe to grow. And then you score for color change, which indicates that that microbe, in fact, grew in that media. Okay, so for here, this is after it's grown. Okay, and so you'll see that here they scored that one as positive. This one, there was no color change, so negative. This one, positive, positive, negative, and so on and so forth. And then you can input all of that data into a computer, and it will compare it to a database, and it will tell you, okay, this is the profile of this particular microbe, like a Pseudomonas or something like that. And you can be fairly confident that you have identified the microbe, and you've also given it lots of tests all at once in a very high throughput way. Okay, so this would be an example of the uninoculated strip. So this is just what the media looks like before any bacteria are in there. And then you have inoculated it with either E. coli or Proteus mirabilis. And you can see that they have vastly different profiles based on their ability to grow in those individual tubes. And that is all saved in a database, and you can compare it to that. So then you can tell, all right, I'm dealing with E. coli, I'm not dealing with Proteus, or vice versa. Okay, so this is, I do not want you to memorize this. I just want to show you that this is how one might go about trying to understand what you're dealing with. And it's basically, like I said, a dichotomous key. So you do a test and you say, was it negative or positive? If it was a positive, you're going to go down this route. You do another test. Is it negative or positive? And then if it was positive, you go down another route until you get to an endpoint. Okay? So for example, you have a gram-negative rod. All right, well, that doesn't tell you much. Okay? So at least you know it's not gram-positive. Then, can it utilize glucose? Yes, it can. Can it utilize lactose? No, it can't. Does it make hydrogen sulfide? No, it doesn't. Can it use citrate? Yes, it can. And then you get down to here to this bacteria. And so it just tells you, it guides you along the way through the series of choices and tests that you need to do. And that gives you an idea of what you're dealing with. And that's all based on biochemistry. Okay, so that is how a bacteria behaves when given a certain substrate, or is it making a specific enzyme that you can test for, so on and so forth. Okay, so you can do this in a very high throughput way. So this is a 96 well plate. An example would be a Biolog machine, which is a proprietary company that makes these plates. And you inoculate your bacteria into these plates, and then you get color changes in all the different media types here. And then you put that into this machine, and it reads the plate, and then it compares it to a database, and it tells you what you're dealing with in terms of potential microbial infections. Now, this is very important that you use standardized methods, because if you inoculate the wrong amount of cells, or if you inoculate old cells, or if you inoculate cells that are at the wrong growth phase, you can get erroneous results, and that can cause you to misdiagnose, and so you don't want that to happen. Here are some other specific tests for identifying gram-negative non-enteric bacteria. So what does it mean? I haven't defined this really. Enteric versus non-enteric. Okay, so enteric is kind of a catch-all term for things that are in your gut, okay, and belong to the Enterobacteriaceae. So E. coli, Salmonella, all of these bacteria that are what we call enterics, all right? There are others that are non-enteric, so they might live on your skin, and they're not related to E. coli. They're not in the Enterobacteriaceae. Those are considered non-enterics. So an example would be Haemophilus influenzae or Neisseria meningitis, right? So you can do this test where this bacteria, we know, needs two factors to grow. And so you can have this segmented plate, right? So you have blood agar in one section, you have blood agar that has the X factor, this agar has a V factor, but it needs X and V to grow. So you can do this series of tests, and if it only grows here, you can say, all right, this is probably Haemophilus influenzae. Neisseria meningitis produces oxidase, so it's positive for the oxidase test. And it turns this sort of purple color on the media if oxidase is present. And so then you can say, all right, yes, I'm dealing with Neisseria meningitis. Gram-positives, just see. Okay, I'll go through gram-positives and then we'll end for the day. So gram-positives, there are another series or battery of tests that you can do. So you can put nalidixic acid in the plate. That kills off all the gram-negatives, but the gram-positives are generally resistant to that. You can do a catalase test. So this is an enzyme that converts peroxide to oxygen and water. Very conveniently, it produces bubbles. Okay, so if you have bubbling, when you give this bacteria peroxide, you can say, all right, this is catalase positive. We already talked about hemolysin. So this is the production of those toxins that lyse red blood cells. You can see that in a petri dish. If you look around, you'll see a halo, if you plate it on blood agar, of lysed red blood cells. You can also do a coagulase test, which you put it in a specific media and it coagulates into this big clump, and that would be coagulase positive. And then you have another series of dichotomous E tests for gram-positives that are different than those for gram-negatives, but the whole idea is the same, okay? So you would plate it on a specific agar, like blood agar. Does it grow? If it grows, you do a gram stain, it's gram-positive. Do a catalase test, it was catalase negative. Then you do a hemolysin test and go, oh, it's hemolysin plus. Is it resistant to bacitracin? Yes. Then you have Streptococcus pyogenes as your example. Okay, so we'll leave off there and catch up on Friday.